So, dear participants, welcome to the fifth day of the STC and the last day of the STC. And today we are going to discuss two very important tools. First is to be discussed by Dr. Amit Kamra, and that tool name is Orange. And second tool which we'll be discussing today is NS3, which is for WSM. So this first session is taken by Dr. Amit Kamra. So I welcome Dr. Amit Kamra to take this session. And Dr. Amit Kamra is working as Assistant Professor in Department of Information Technology at Guru Nanak Dev Engineering College, Ludhiana. He has teaching experience of more than 20 years. He has done his BTech from PTU in 2001, then MTech in Information Technology in 2004 from Punjabi University, Patiala. And Dr. Amit did his PhD in Computer Science from Slide Longowal in 2014. His areas of interest are data mining, entrepreneur skills, digital marketing, finance management, machine learning, virtual reality, image processing, and outcome-based education. He is also PhD coordinator and Institute MBA convener at GNDEC Ludhiana. He has recently received Best Teacher Award by ISTE in December 2020. And Dr. Amit Kamra has given a number of expert talks in various institutes of repute. He has received grants of more than 25 lakhs from funding agencies like UGC, PTU to organize various activities. He has published papers in journals of repute with indexing in SCI and Scopus. He is also reviewer of reputed journals published by Springer or Elsevier. He has guided 31 MTech theses and 2 PhD theses till date, and 2 students are working under him for completing their PhD. Introduction, I invite Dr. Amit Kamra to take up today's session and start. Dr. Amit, over to you, please.
Thank you, Dr. Saab, for a nice and a crisp introduction of me. Good morning to my dear friends. And today is a very nice day at Ludhiana. The weather is very nice one. The monsoon season has started, and I hope the weather at your place also might be good now. And I hope all of the India will be flourished with this monsoon rains in the coming days. Why I'm talking of monsoon to nowadays, because we have already bear the brunt of corona from the last few months, and the hot weather might have intensified that fact. So a good weather and a good lecture may try to provide you some relief in terms of knowledge and in terms of some comfort to the body also. So once again, good morning to all of you. And I'm looking at the number of the participant list. Many are from the South India, and I hope my accent will be comfortable with all of you.
No problem, sir.
Yeah. Sir, if there is some bit of sound issue also, because I'm sitting in my staff room, and if there is some sound issue, please do tell. Okay. And lecture is going to be very interesting one, because this is not any theoretical lecture. Just wait for 1 minute. I'm going to close my office door because there is some noise outside. Please just wait for 1 minute. Okay, I'm back now. I'm sorry, there was a bit of noise my joining staff room. So this lecture is going to be a very interesting one today, because this is not a traditional theoretical sort of lecture. And after listening to this lecture for the next 1.5 hour, I hope you will get some good idea about a tool called Orange today, because nowadays there are a lot of emerging areas, and one of the best emerging area which can really help our stakeholders is this machine learning, data mining. We are always talking about data. Okay? So think of a tool, some sort of tool that can really help to optimize that data, that can really help to visualize that data. आप लोगों ने MATLAB के बारे में नाम सुना होगा. You might have heard about MATLAB. MATLAB is a paid tool, and when I was invited for this talk by Dr. Dhaliwal, I just shared my view that Dr. Sabha, can I discuss some simulator in the lecture? Then he told that we want open source tool because the theme of the FDP is some open source tool. Then we discussed and finalized that I should discuss something about a new tool called Orange, and Orange has similar sort of toolboxes, uh, just like similar like MATLAB. The good thing is that this tool is a freeware software, and you doesn't have to pay a single penny to buy this software. So the topic of the theme is today is data visualization and data optimization. Uh, and I hope with my practical demonstration, which I'm going to show you during the course of the lecture, you people will be easily customized to learn this tool at your end also. Okay. So please, I beg your pardon if because I have to shift between the both lecture as well as the demo part. So there will be small time lag between these two things. तो अगर कोई दिक्कत आए तो आप बीच में पूछ सकते हैं, ठीक है. And in fact, I will be also asking you many questions. So don't treat कि मैं ही अकेला बोलता रहूँगा. Because otherwise the lecture will become monotonous. So whenever I ask you something, please be prompt and unmute yourself and discuss with me. It is not a just like question answer session. It's just a discussion forum only. Okay. So almost every area of research require visualization and optimization of the data. I hope many of you are PhD faculty members and many of you might be doing PhD. So this tool can really help to optimize the data. Either you are from electrical, I hope many of the participants are from the electronics and the computer and the electrical background or the IT background people. So this tool is really a boon for those people. There are certain tools that can really bring out the difficulties from this task of visualizing the optimization of the data. Now, there is one problem here that most of the people they do not want to learn coding. ठीक है, coding बहुत लोगों को पसंद नहीं होती है. That's why there are certain tools that can act as an alternative to this coding component. So the solution here is Orange. Today I will discuss you to another GUI based tool called Orange. Orange is an open source data visualization and analysis tool. With the data mining, with the machine learning is done through Python scripting. But डरने की बात नहीं है. No need to fear about. We are not going to learn about Python. हम Python नहीं पढ़ेंगे. हाँ, Orange बनाया Python में गया है. ये I just want to share with you. Whenever we heard the name Orange, it gives first impression that it is the name of a fruit. Obviously, orange is the name of a fruit, but here we are going to discuss about a tool. This is a small icon symbol of the orange, the person wearing a black specs. And this tool is really very useful for the persons working in the area of image processing, bioinformatics, text mining. It has around 15 toolboxes, and I am given 90 minutes for my lecture today, and I will try to cover 3 to 4 toolboxes, an idea of 3 to 4 toolboxes. The rest of the toolboxes depends upon your area of expertise or area of the research. So, आपको आज चलाना आ जाएगा ये tool. You will people be able to how to operate this tool, and with the examples I'm going to discuss with you in the due course of time, you will get a good idea about that how the results are being obtained. So this tool is basically beginners who want to visualize the patterns. आपने कभी PhD report में भी graphs बनाए होंगे. I just ask you one thing. Let me ask my participant. Sandeep Kumar ji. Sandeep ji, can you able to listen my voice? Sandeep ji. Sandeep Kumar ji. I think Mohammed Siraj ji. Mohammed Siraj ji.
Yes, sir.
Mohammed ji, do you know any sort of graphs which we use in our PhD or MTech thesis report जो हमारे students लगाते हैं? Any sort of graphs?
Graph?
For graph या visual diagrams जो हम for the data जो results के लिए बनाए जाते हैं. There are एक graph था जिसे bar charts बोलते हैं. Bar charts. Simply a Venn diagram. We might have studied in our 10th class. Venn diagram. So can you give some other idea of what sort of graphs? Any participant can tell me what sort of graphs we or diagram which we use to visualize the data. जब आपको pie chart. Exactly.
Histogram.
Histogram बड़ा अच्छा है.
Bundle diagram, sir. Dot matrix type.
Bilkul, exactly. Thank you. All of you can mute yourself. Thank you, my dear friends. But these are very common diagrams which we use traditionally from the last many years. Madam ne naam diya histogram, pie chart, Venn diagram. But there are around 20 to 30 types of another diagram which are very useful when we plot corresponding to our data. The only thing you have to find out that which diagram is suitable for your research work. In fact, when I did my PhD, I was asked by my supervisor to plot box plot. I hope you might have heard the name box plot. ठीक है तो बॉक्स प्लॉट बनाए पढ़ते थे डिस्ट्रीब्यूशन डायग्राम वेन डायग्राम स्कैटर डायग्राम sieve diagram heat maps all of these names are not familiar with you and if you try to incorporate any of these diagram in your report although it must be relevant to your research work ये नहीं है कि हमने सिर्फ बनाने के लिए बना देना है but you have to feel that how the diagram analysis is going to really help you in your research work. So the good thing about Orange is that it is able to provide a huge number of the visualization patterns which we normally do not know in our normal life. तो जैसे शांता मैम एंड प्राजना मैम एंड संजय जी ने बताया अभी हिस्टोग्राम वेन डायग्राम और बार चार्ट पाई चार्ट दीज आर वेरी कॉमन नेम्स जनरली ये एक्सेल में बन जाते हैं बट यहाँ पर आपको कुछ नहीं करना सिर्फ एक छोटा सा बटन दबाना है तो यू विल गेट ए ह्यूज नंबर ऑफ ऑप्शंस तो डिपेंडिंग अपॉन योर चॉइस यू आप अपना ग्राफ बनाइए विजुलाइज कीजिए आई हैव इन रिव्यू ऑफ द मेनी जर्नल्स ऑल्सो तो जर्नल्स में हमें ना एक कहा जाता है कि उस पेपर को एक्सेप्ट करने के चांसेस बढ़ जाते हैं इन विच दे द रिजल्ट्स आर बीइंग एक्सप्लेन थ्रू द डायग्राम एंड द टेबल्स so it is better to predict the diagram okay Murugan ji I will speak in continuous in English so it is better to predict to analyze the diagram of the results in form of tables and graphs only so let's come back to the presentation again yeah I will communicate in English only. So what basically is Orange here? So Orange is an open source component-based visual programming software package. Whatever the applications I am writing here, I will try to show you all those things practically also. So don't need to worry here. Orange is used for data visualization. Just I initially told that there are a number of diagrams which we can make in Orange. So I will be showing you the diagrams also. I will be showing you image processing toolbox also today. What sort of image processing we can do here. Bioinformatics is not my cup of tea, although some of my friends might be working in the area of bioinformatics, but bioinformatics processing is also possible here. Machine learning we will be doing here, data mining we will be doing here, and data analysis will be, I will be showing you through some examples also. So Orange, the initially in the start of my lecture I told that you need to do some coding the only thing is to you have to uh, good idea of the components of the orange so the components of the orange they are called widgets widgets are just like something some small buttons which have some predefined functions in them okay so what these widgets can do here, widgets they can provide data visualization. Which widgets can do subset selection. I will be showing you subset selection also. Widgets can do pre-processing also. Maybe I will be showing this also. Widgets can help in predictive modeling. We will be doing all those things practically today. So when we do a certain program, we write an algorithm and write lines of code here. So the functioning which is done in being orange is called creating a workflow. So suppose at the end of the lecture, I will be giving you one small assignment so that you can practice it later on. But in that assignment, you have to download Orange and you have to create the workflow and then visualize the results and analyze the other results. So the thing is that you don't need to do program. You have to create the workflow. You have to enter your data and then you have to visualize that data. You have to pre-process that data. You have to do selection of the data. You have to classify the data. You have to make some clusters also. Okay. So the best part of the Orange is that it is an open source tool, and this tool is undoubtedly a boon for the people who are doing PhD or masters in the area of CSC, EC, or IT. One can modify the algorithm also as per the requirement, and you can just enter your data and you can get the results. The only thing is that you must be fully customized to the basics of the machine learning or the basics of the image processing This is just a tool for all those fields. So I'm not going to discuss today about machine learning. I'm not going to discuss today about bioinformatics or image processing. We are just going to discuss that how that this tool can be used in those fields. Please, my dear friends, my screen, I having a full screen, so I'm not going to see your comments in the chat boxes. Otherwise, I will get distracted. But if you have any doubt, you can unmute yourself, or you can ask in the chat box, and I will reply at the last of the lecture. But in between, also, you can ask anything. So we are slowly proceeding towards the demonstration of this lecture now. But before that, let's have some more idea about this tool. This tool is not just meant for engineering people also. This tool has a assistance for the person working in remote sensing, person working in geographical location, person working in the field of networks, person for working in the area of text mining. So it's just like simply installing a game and then you have to play a game. So you this working on Orange is a really a very fun sort of mechanism. Okay, so you don't need to learn any programming language also. The only thing which you know is your domain in which you are working. It support around 10 to 15 domains. I will be showing you all those domains also. So if that is your domain, you need to know about those particular widgets and their working. Although I am not customized to all the domains because my area is image processing and a bit of virtual reality, as Dr. Thaliwal told in the initial start of the lecture. Every person doesn't know all those things. So it depends upon which domain you are working, and Orange, if that domain is already supported by the Orange, it's a very nice opportunity to you people that you can use its results, which are very easily workable and very easily retrievable, and you can easily get the analysis and get the printouts of those results, and you can paste it in your thesis report or your research paper. Okay, means you should know which algorithm or which widget should be used in a particular scenario. So, with our experience and the practicing of this tool, we will be getting a good idea about the working of the tools. Okay, so I cannot explain all the widgets today. There are around, I hope, there are 500 widgets in this, the complete package of Orange, and I have installed around 200 widgets in this software, whichever I need now. And out of 200, I will try to show you around 50 to 60 widgets. So 1/10 of the widgets I will try to show you today. Orange is a very smart tool. So you will definitely fall in love with this tool when you experience that it is very flexible and it provides a very visual environment. And the good thing about the Orange is that you have to make a connection between the widgets. And if the connection is wrong, Orange itself automatically guess that the connection is wrong, and it will never provide you a wrong connection. So there is no possibility that the result will be wrong. Only the output will be lesser accurate or inaccurate. It doesn't matter. It depends on that depends on the data, but the in connection, the wrong connection of the widgets is not possible. I will try to make a wrong connection, and you can just note it that Orange doesn't support the wrong connection also. Okay. So you can have a screenshot of this this slide. This is a very important one. Orange was developed by a company called Biolabs. It is an open source community. The important thing is that when you have to download this Orange in your, it the capacity it occupies is 480 megabytes. So it takes 5 to 10 minutes to download the Orange, but you have to make sure that your Windows is 32-bit or Windows is 64-bit. It comes with the Windows version. Apple and the Unix version are also there, but I'm today using the this Windows version. So this is a link also I have given here. Although the link I mentioned is 3.3.2 27.1 this version, but yesterday only I updated, and the new version is now 29.3. So you can imagine how many versions of the Orange has came till now. They keep on upgrading their uh, toolboxes every time. I'm a member of this community, this BioLab community, and we keep on providing some workflow, just like the MATLAB has its community, mathworks.com. So people do write some code and share to the persons working in this domain. Similar sort of community is developed by the BioLabs also. The people working in the Orange community they can share their workflow, they can share their experience, they can demand for the new toolboxes with this Orange community keeps on creating in this toolbox. I started learning Orange from the bio labs in 2018, and at that time only 3 to 4 toolboxes were there. And till today, I have around 7 to 8 toolboxes in my laptop. There are around 15 toolboxes now, but 5 to 6 I have already installed in my PC, and I'm quite customized to all of them. I will be showing you those toolboxes also now today. The good thing about the Orange is that when you're going to install the Orange, it will install its default version also only. So if you want to install the image processing toolbox or a bioinformatics toolbox or text mining toolbox, you have to do add-ons. I will be showing you how to do add-ons also. So that you need to install in addition to the default version. Okay. So whenever you want to, please, this is one sort of error that comes if your Windows is not updated. So you can install either you update your Windows, because some of my friends might be using the older version of the Windows, or the Windows might not be updated. So you have to download this file from the Google libbz2 is the DLL file, something concerned with Windows only, so not our domain here. You have to copy this file and save in the Windows directory in your C drive, and then you can install it. Yes, ma'am. So if the operating system is fully updated, then you will not get any error. Okay. So this is the picture I'm going to show here. Anybody can acknowledge me? Is this visible?
Yes, sir,
Can you
visible.
go back to the previous slide?
Yes, sir.
Yes, Chali.
Previous one.
Okay. I will be giving the presentation to Dr. Dhaliwal, don't worry. So you can, this is not my copyright item. These are slides which I have learned from this biolabs only. So don't take any tension of those slides. If you don't want to listen to the lecture again, you can have the screenshot of all those things. But my dear friend, this error will not come definitely. I have just thought if the operating system is not fully updated, then only you will get this error. Okay. So just I need help of some person. Sanjay ji, please keep on assisting me whenever I ask something. Sanjay ji, Sandeep ji, or Kulvinder Pannu. Okay. So please, is this visible? This demo?
Yes, sir.
Okay. Just I'm going to minimize this. Are you able? Just see here. This is the symbol of the orange. Okay. Kulvinder ji, who is speaking now? This is the symbol of the orange. Okay. It's being installed here now. Just see here options. You have to go on add-ons. Okay. It is going to retrieve the package list now. It will take 2 minutes time. My net speed is good. Somebody can see here now. Orange Associate Bioinformatics Educational Explain Geo Image Analytics. What's his name? Shanta ji.
Yes, sir. Yes, sir.
Ma'am, can you see here? Can you see here? These are the toolboxes here.
Yes, sir. We can see.
Okay, and there are certain toolboxes which are already being installed, so they are displayed through this tick button, Geo and Image Analytics. So somebody is working in computer networks domain. So this is his area. I didn't work in networks, so I never installed. Prototypes is something concerned with creating prototypes. This single cell is concerned with biology, I think, bioinformatics. Spectroscopy is a cup of tea for the persons of the chemistry domain. Text mining, I'm working in text mining, and I will showing you text mining today. Okay. Time series also, I believe time series is used for the COVID data analysis. So although I will not be showing you the demo here due to the paucity of time. And survival analysis, you can see here around a few tools I have already installed it, and other things you can install it whenever you require. So I'm just going to close it. Okay, because I'm going to install it, then it will ask for the restart again. So let's come back to the presentation again. So this is you can see here, these are additional widgets. So whichever you require, you can install it for your work. So it depends upon area of your expertise. In this Orange, when they started developing the Orange, there were only 2 to 3 add-ons, but right now there are 11 to 12 add-ons. So the community keeps on working to just like MATLAB has a lot of toolboxes, so Orange has a also trying to incorporate many of the toolboxes. MATLAB cost is very high; it is a costly software, and the main advantage of Orange is that it's an open source tool. And if you get the idea of the working, the results can be really helpful to you, and the results which you attach in your report based on the visual patterns or some classifier based on the machine learning are really going to give you very good results in a very less span of time and with very high accuracy. Okay. In fact, I completed my PhD in 2014 and I was not knowing Orange at that time. I used to work in MATLAB, and in MATLAB I have to do that, create script files also. Although I'm not going to say which is better. Both have the different aspects, but in when you are working in MATLAB, you have to create fig file, you have to create script file, you have to do the coding. All those things are not being done in Orange. So when you get the idea about the working, all the results will be very similar what we obtain in MATLAB and what we are getting in Orange. Okay. I just saw this all those things in the last slide only. So let's start Orange now. I have already installed Orange, so I have given you that link also. But please make sure just check your Windows is 32-bit or 64-bit. Otherwise, if you install the incompatible version, the Orange will not work. So whenever you start a Orange, you will see a welcome screen. I have disabled it already. You can exit that welcome screen and Orange start with a black canvas. This is Orange again. You can see here, this is the data. I'm just scrolling it down. You can see the number of widgets here. These are widgets: file, CSV, data sets, SQL table, data table, paint data, aggregate columns. Okay, let's scroll it down. Is there some time lag or is it visible comfortably? Manisha, is it visible comfortably?
Yes, sir.
Sir,
Okay.
yes, sir.
Okay, thank you. So you just see the visualizing pattern, tree viewer, blocks plot. See the names now, different names you might have never heard in your life. Violin plot, scatter plot, line plot, bar plot you might have heard it. Sieve diagram, mosaic display, free viz, linear projection, other is rad viz, heat map. Heat map I will be showing you. It's not possible to show the demo of all those diagrams because there are around 20 diagrams. Nomogram, CNN viewer, Pythagorean tree. So depending upon your research area and what sort of results you can retrieve from these diagrams, you need to incorporate in your research paper or your PhD or MTech thesis. Okay, so it doesn't matter. There are 20 diagrams, and every diagram is compatible with your research work. So I just want to give you a small warning here that don't go for visualizing every diagram here. Okay, just scroll down. This is the models here: CNN rule, Knn classifier, tree classifier, random classifier, support vector machine. You might be surprised to know here that there are around 20 to 25 types of classifier here. And the important thing is that you must have the theoretical understanding of these classifier, because the values which are being input to the classifier will depend that how that classifier is fine tuned. Suppose if you're using Knn classifier, you have to give the value of the K, because unless and until you don't have the knowledge of that particular classifier, it doesn't make sense. You will get some results and you will not be able to analyze those results. Just come down here, evaluate, testing and score, cross validation. You just, I'm showing you after some time. Confusion metrics, ROC analysis. I plotted confusion metrics and ROC analysis in my report with some coding also. See the unsupervised classifier, hierarchical clustering, K means, manifold, P, principal component analysis, distance vector, distance transformation. Now come the image analytics tool, image analysis tool. Come down, come is the time series, Yahoo Finance, interpolate, moving transform. I'm not knowing all those widgets. Okay, so being an expert in any domain, it requires a lot of time, because my area of domain is image processing and text mining only. So I will try to show you those examples. But if you are working on these domains, definitely with some help uh, from the bio labs, you can easily be accustomed to all those tools. Text mining, I will be showing you. Text mining is a very popular field nowadays. You might have heard about the sentiment analysis on Twitter. The Twitter sentiment analysis, it is a very important aspect that nowadays the government also try to find out that how many people speak in the favor of a policy of a government and how many people speak not in the favor of a government. That term is called sentiment analysis. And with the help of these tools, we can easily do the text mining also. I will showing you all those things today. So just see here, there are around 200 widgets I have shown you. And if I installed all those add-ons now, there are around 500 widgets. Okay. Is it clear what are widgets? These are small components are called widgets. Anybody please acknowledge me in between? Uh, Shantaji ma'am.
Yes, sir.
Okay, Farha Pathanji.
Yes,
Farha Pathanji,
yes, sir. Yes, sir.
is it clear what are these widgets? Okay,
Yes,
Yes,
sir.
sir. Yes, sir.
let's come back. So these orange start with the black canvas. This is a black canvas. I just shared in the morning. I just shared in the sorry in the start of my lecture that you will feel that you are just doing a drawing sort of. So this is the canvas part. Just take. I beg your pardon for 1 minute. पूरा lecture लग गया है, मुझे further करना है। So this is the canvas part. We have all have kids at our home and they do the drawing on the canvas. So you people will be also doing drawing on the canvas, but that drawing will be the technical drawing today. Okay. So whenever this all this magic will be done on the canvas, and for the just like our kids, they need some good drawing colors. I have a younger daughter of 10 years age, and she is very fond of doing this painting. And nowadays, during this COVID period, the children are very free at home, and they keep on asking that Papa, please bring some stationery items. They keep on telling me, I want some watercolor, some crayons color, some variety of colors. They have different options available in colors here. So you also people will be having different widgets available, and that widget will be serving some different purpose also. So let's do here. This is a welcome screen. I have already disabled it, so it doesn't matter if you install the audio for the first time. This screen, just click on the show at the startup, and it will not going to disturb you again. Okay. So let's start the orange now. So what are these widgets? Widgets are the computational units of the orange. Whatever the widgets I have shown you just now, I will be showing you working of all widgets now. Since this lecture is of 1.5 an hour, I've also taken the snapshots of those lectures because if you want to practice the workflow later down the stage, you can just see what I have practiced it, and then you can practice it on your PC, which will give you a good idea about the working of those workflow and the working of those widgets also. So these widgets are used to read the data, they are used to process the data, they are used to visualize the data. We will start whatever things I have mentioned in the start of my lecture, that we will be doing pre-processing, we will be doing visualization, we will be doing clustering, all those things we will be showing you here. Okay, so let's start here. This is the file widget. So how is it come here? You have to just click here, single click, and you have to click on the file, just double click it. Now you can see here, there are certain default tables stored in it. Orange has some default database with it to show some practice examples also. If you have your data, you can also browse it. Okay. Data is something, some numeric values. See, computer, we have a computer, and whenever we process with computer, we are just given some alphanumeric values and some text only. So I have opened trying to open one database called heart disease tab. There is another thing URL written here, and this URL is pasted here, so please ignore it. So you can see here, I open my PC and just browse it. This is a default some database which I have chosen now, heart disease data tab. Just minimize it, and then come back to the slide again. Most data analysis occur with a file widget. I just click on the file widget. It has appeared in the canvas part. And when I double click the file, you can select any of the data set. So Orange itself has a lot of data sets, but for simplicity, we are loading one data set called heart disease data set. Heart disease data set is a data set which is comprising of the number of samples of human being with some biological values. Okay. And on the basis of this, we will be doing some analysis on this data set. So you can see here, this is a same screenshot which I got here in this result itself. So the next thing is you have to click on the data table. You have to connect the file to the data table. And now click here on the data table. Somebody, please, you can see here. Farhaji, you can see here. This is the diameter, age,
Yes, sir.
chest,
Yes, sir.
chest pain,
Yes, sir.
static blood pressure, cholesterol, blood sugar, rest EG, maximum HR is some biological parameter. I'm not from a biology domain, so I'm not fully customized to all those parameters. We
Heart
just
rate.
yeah, okay. Thank you. Exercise induced angina, ST by exercise, slope, peak over excess ST. So don't worry about these all values here. These are just biological parameters of a body by which somebody can predict the type of the chest pain. It is typical angina, asymptomatic, or something another is called non-anginal. So we don't need to worry about that. What are these biological parameters? Because we are basically doing machine learning now. So let's come back to the presentation now. So this herd disease data set, when I open the herd disease data set in the orange directory, we click on that data table icon and it appear on the canvas, and we are able to see something here. Now the good thing is that I have connected the file widget with the data table here. Just see here. When I connected the file with the data table here, they both start communicating each other. So connecting a one widget to another widget will lead to some results also. And just here, I'm doing something weird here now. I have connected data table. I have connected here. Farhaji, I told you starting the lecture one thing, that is, it is a smart tool. Somebody please acknowledge me why that it doesn't happen. I'm trying to connect it, but it's not doing connection.
So if we are doing a wrong connection,
Ushaji, thank you very much. So that means Orange itself detects that you are doing something wrong. Okay? So there is no scope of error. The only the thing is that your results will be inaccurate. If you try to make a wrong connection, the Orange will not allow you. Okay? So now we are connecting to the data table now. And now come to the next part here. This is again table description shown here. Just see here, if you want to plot something out of this, you have to click on the data table. Right here, scatter plot. Now just see here. This is the scatter plot with some axes here, age, gender, cholesterol. You can see a fantastic thing happening here. Changing the parameters, you are getting it very nice visual patterns. Don't worry about that's how this graph is being done. There are some age and maximum HR, some sort of this biological values are being description. On the x-axis we have the maximum HR, and the x-axis we have the age, and on this side we have having the HR. Somebody's mic is on. Please, can you unmute yourself? Sorry, mute yourself. Otherwise, there is some. Muruganji and Sanjay Puriji, please, if possible, can you mute yourself?
I am
Very
already
small.
muted, sir.
नहीं संजय जी अच्छा आपके you have logging from the two sides, some sort of disturbance. I welcome if you have doubt, but that small noise cause some problem. Okay. Let's plot again. Just once again. Okay, this is a scatter plot here. Okay, you can see the scatter plot now. So this is a very nice visual analysis. So again, somebody's mic is again on. So these are a please, Murugan ji, Dhaliwal sahab, please tell Mr. K. Murugan to mute yourself.
Mr. K. Murugesan, kindly mute your mic, please.
सब मुरुगन जी कर लीजिए please. Thank you, sir. Thank you. Please don't mind because this creates a bit of disturbance on this platform is very causing some disturbance. Otherwise, if you have doubt, you can disturb me anytime. Okay. So let's come back to again. So these are the types of plots you can create in the orange. One is called box plot, sieve diagram, scatter plot. Initially, at the start of the lecture, Sanjay ji and other friends have told about certain names. So depending on the requirement in your research work, you can select the type of plot suitable for your research work. Because when you get some result, when you get some data and you provide some tabular result, and instead of providing the tabular result, you try to make it some plot with some parameters, it will create a very good impact. And if you do some analysis of those result and explain that graphical result in a very nice manner, the chances of acceptance of the research paper or your thesis will be very nice one. So because unless and until some graphical results are added in your report, they creates a very significant impact. So they are some sort of unique plots, but the only thing is you have to find out which plot is suitable for your research work. There are around 20 to 40 diagrams which you can plot in the this orange. I've just shown you one only, scatter plot. So let's see again here. We're going to create the data flow here. Initially, at the start of my lecture, I told that when we do the coding, we make programs, and when we do the analysis in Orange, the term is called workflow. So please make sure whenever you get some assignment or you should do some work in the Orange, you have to create a workflow. And if somebody of you is teaching this machine learning or data mining in your college at your BTech or diploma level also, you can incorporate these tools in your curriculum also, because this is a very simple tool and the students will very easily understand this, and they will very love to watch and work on this, because some people avoid doing coding. There are certain students who likes some working on GUI also. This can really create a very good interest among our stakeholders also. So what we have done here, we have simply loaded the data that herd data set, and this herd data set you can see here. Come back to the table again. Data table has 303 instances. It has 13 features, and you just scroll it down. These are the 13 features. Okay, and if this is the result here, and if you find that this result, this part is the overlapped one. Okay, this part is overlapped one, then you can connect scatter plot with another scatter plot to have a better and detailed visualization. Now just see the difference between these two plots. This is scatter plot between age and maximum HR, and this is again age and maximum HR. This is the maximum HR. Now this is the scatter plot of the already selected portion. This term is called subset selection. Okay, I told in my start of my lecture that you will be able to visualize the result. You will be able to do the subset selection, and this term is called subset selection. So the widgets they communicate with each other. Connecting, when I connected this scatter plot with another scatter plot, and I selected some particular data set out of the complete data set, we get another scatter plot with some greater and very better visualizing results. This is subset selection. So just see another diagram now. Again, we connect a data table to with the. You don't have to do with a lot of hard work. Only simple drawing only. Just drag here and write the name of the diagram. Distributions. This is another virtual diagram. Distribution diagram. That how these. Now it's try to grab this picture and add in your results in your PhD or your research paper. It will really going to give a very good impact on the visualization when the reviewer check your paper. See this all those things, and when we make in Excel, it takes a lot of time to make this graph, and just clicking on these buttons with some x-axis and y-axis value, you're getting a number of the results. Don't worry about these values. These are values are those depicted in the table. So some values are higher and some are lesser. So accordingly, their values are changing here. So this is a distribution plot. This is also a appropriate for this such sort of problem. Although when we connect data table here, when you connect data table to, you can find out the number of diagrams here. Scatter plot. Just write on one diagram more. Line plot. Now see here. Can you see here, Farhadi? Can you see here? This icon comes here.
Yes, sir.
This when this sort of icon comes, it shows that some values are being missed, or automatically the orange is going to tell you the results which you're going to get now will not be appropriate one, although it has make the connection also. But uh, the thing is that the results which you have retrieved is not very optimal ones. Now see here, what is line plot here? This doesn't make a very good sense. All the lines are merged one, and some useful analysis cannot be made out from this diagram. So you have to choose the diagram depending upon your data values, or some sort of analysis have to be done. So a small kid can also do all those things, but a researcher or a PhD professor or an engineer uh, has those skill to do the analysis of that data based upon the type of the graph he has chosen. So it will depend upon your skill or expertise that which sort of distribution the graph is suitable for your result. Okay, so let's come back again. So this is a distribution widget. Okay, this is another I have shown in that slide also. I have added a slide just for you people, because after that it is not easy to understand all those things, because when you watch the slides again, you can easily practice it. And after reading this lecture, you will be able to learn about 25% of the orange. I bet you people will learn. And rest of 75% takes some practice, and in a span of 2 to 3 weeks, you can be fully customized to practice on one tool of your research area. Okay. So learning a complete orange package will take a lot of time, and it's not possible because we are not expert in all the domain. I don't have any idea about the ABC of the bioinformatics, because that subject was elective in my BTech curriculum, and my research area was image processing. So I don't have that genome prototypes. One of my colleague, his PhD in bioinformatics, and he used to use these terms whenever his PhD or said at MTech student Viva voice is being held in our department. So I always used to sit quietly because I don't have idea of all those things. And similarly, if you have a good idea about your toolbox or your domain, then only you can become an expert in those domains. Okay? So this is category again of the same data set. We do some sort of subset selection, and when the subset selection is done, you get a better analysis of the result. Okay? Because it is data. Orange Box is all about data visualization. Okay. So the good thing is that the changes in one widget, they are communicated immediately to the next workflow. So this complete diagram is called workflow. Okay, you can either save it. You can, when you save it, you have to name it by extension name OWS. Okay, you can save it for the later use also. You can discard it anytime whenever you don't feel. Okay. What are the methods to add widgets in the orange? Let's come back again. Let's delete it. You have to drag it and delete it. How can you add widget? You can add them. You can double click them. Okay. You can come to this file menu and just click it. Either write the name of the widget here. Then also, is it possible? Or you have to find out the scatter plot. Where is the scatter plot here? Just scroll down this. This is scatter plot that we have to find out. The easiest task is just to have just the write the name here, line diagram. Okay, whatever the widgets, these are stored in this visualized domain. Okay, I'm going to write out something called bag of words. Now this bag of words widget, you have to find out. It takes a lot of time, and this bag of words widget is in the text mining widget. This is a bag of words, so it takes a lot of time here to find out something. And please see here, this is the data. This is another thing which we have seen here, red line. Sanjay ji, here, can you see here? This is a red line here. Otherwise, the diagram was black
Yes,
lines
sir.
every time. See here?
Yes, sir. It is red.
This is a red line, and this means that this bag of words widget is compatible with some another widget of his domain only. This bag of widget is compatible. It is telling that data corpus. This file widget is for the general image, a general task, but when we have to process on the bag of words, it is telling that you have to connect with the corpus widget. So just here, I'm clicking a corpus widget, and I'm connecting to the corpus view. I will showing you. Just wait for 10 to 15 minutes. This widget is also coming after some time. We will discuss at that time. So these are different ways to connect the widget. If you feel this space is lesser, you can see here view expand tool deck. Your canvas will be bigger one. If your kid asks that Papa, I don't want A4 sheet, get me A3 sheets. So this is the A3 sheet, bigger size for you, and that was a A4 sheet. Click here, expand tool deck. Your tool will be, your canvas will be a smaller one. Okay. Just to tell you something about this. So you can also tell the orange what kind of data can be sent from one widget to another. So let's see some practical aspect again here. Just going to something. Let's shift this, skip this. Okay, loading and saving your data I've already told here. The Orange supports Orange. The data which we can enter into the Orange is of generally three types. It can read the XLS file, it can read the tab file, and it can read the CSV file. So if you have some data in your laptop or in your PC, it should be in either in these three forms. And I hope most of the data in our, whenever we work on some algorithms, is generally in the numeric form only. And you please save it in the XLS or CSV or tab form. Then it is easily to work with Orange. Okay, so it's not a big task here. The good thing about Orange is थोड़ा सा magic करते हैं. Let's try to do some magic now. I ask you one question here. Is anybody doing some PhD from the participant? There are around 87 participants. Can you, Bharti ji, are you a PhD scholar or you're doing PhD? Dhirender ji?
Yes, sir. Yes, sir. I am doing
Sir,
PhD.
I'm tech. I'm not doing PhD, sir.
चलिए भारती जी कभी या धीरेन्द्र जी have you ever felt sometime some sort of problem that you didn't get some database कभी आई
Yes, sir.
दिक्कत so Dhirender ji and Bharti ji, we are doing one small magic here. What I have done till now, we
Okay,
work
sir.
on the already provided data. Now we will do some drawing and we will create some data. Okay, just watch it. There is a widget called paint. सिर्फ painting करनी है. As I told in the start of my lecture, that you have to just do some painting. So there is a paint data tool here. Paint. Click on the paint data. चलिए, this is the painting here. You have to just click randomly here and there. There are two axes. I keep on clicking for 1 minute. Just minimize it. Have the data table. Connect paint data with the data table and just click here. Bharti ji, see here. Dhirender ji, we have got this
Yes,
database.
sir. Yes, sir.
So, can you tell me any limitation here, Dhirender ji? Dhirender ji, any
No,
sort
sir.
of limitation you have felt here?
No, sir.
Bharti ji, anybody, any other friend, you have to find here some certain limitation here in this data?
No, sir.
Ma'am, please speak
So
loudly.
all values are less than 1.
Okay, this is one limitation. One more limitation. Let me ask from other participants also. I just want to see the name. There are a good number of participants here. I hope my lecture is giving some benefits. That's why the many people are. Dalveer Kaur ji. Dalveer Kaur, I think you are from Punjab. Dalveer ji, are you there? Or okay, let me ask Divya Loganathan. If I beg your pardon if I am not spelling the name rightly. Divya ji, Divya Loganathan, I suppose. Anybody else? Surinder ji, Surinder Kumar Gupta ji. Any limitation you feel here?
So the number of data which can be tabulated, and number of decimal places is it 6? I can observe all the data is having 6 decimal places, and the number of data which can be stored may be limited.
No. The earlier somebody I forgot the name who spoke earlier. The
Sir,
one yeah.
one is block right written in typed in blocked and other is somewhat dim.
नहीं sir, that is कोई बात नहीं. That is not a big deal. The important difference, my dear friend, here is that we have the database corresponding only to 2 parameters only. Suppose Bharti ji is working on some database and another disadvantage is that limitation is that was mentioned by one of my friends earlier also, that she told it that the data range is between 0 to 1 only. So who spoke about that? 0 to 1. The range is 0 to 1. That person have a,
Yes, sir.
pardon, sir?
Yes.
Sumitra ji.
Yes, sir.
Sumitra ji, you have pointed rightly. The one limitation was that the data range is between 0 to 1. So if Bharti ji required any data in which the range is required is more, so that this data will not work for her. For this purpose, she have to. This is just a fictitious data, and the other limitation is that we are getting only a two variable data here, x and y axis. So suppose if your data, if you required at least 10 parameters data, and if that data range is required, you need 1 to 100, some sort of fictitious data. If you required that you need around 10 variables, and that variables values can be between 1 to 100, it is not possible right now. So this is a part where Orange community is working every time. I have written them mail also, and we have communicated online with the Orange community members also, that please try to incorporate some widget, some changes in this widget, so that the magic which is done through this paint data widget can be more better for the researchers. So right now you will be getting only the two variable data, and that also in the range of 0 to 1 only. This is the limitation. But anyhow, this is another feature of this Orange database. So using the paint data, we have painted the graph which we want. Now based on this graph, you just get some database. Now this database can be really a miracle for the people who are really short of some data. But the only limitation is that the range is 0 to 1 only. So maybe in the years to come, this widget may have some more better performance. So this is another diagram here. This is a data range here. Okay. So I told one more application called pre-processing. Pre-processing is a very important application of this Orange. This is a fictitious data table which I have created now. What is pre-processing? Practically, can I ask someone? What is pre-processing? Rajni Bhallaji. Rajni Bhallaji, are you from CSC or electronics domain?
So I'm from electronics department.
Okay, Rajni ji, if you are comfortable, can you tell what is pre-processing? General meaning क्या है pre-processing?
Right now I'm not.
Okay, anybody else? It doesn't. Please don't take it as a कोई आपको मैं कोई. It is just a discussion forum, so don't think I'm going to question you anything. Manoj
It is
ji.
the preparation. It is the preparation for doing something. To do some main task, we have to prepare certain things. We have to maybe filter out, maybe modulate, something like that.
Maybe normalize.
That has to be done prior to the main task.
Exactly. So I just speak in my language. Sanjay ji, he has added something to my answer. Sanjay ji, can I say that pre-processing exactly means normalize the whole data? We want to normalize all those things. Pre-processing hota hai, it means clearing all those things. Yeah, exactly. Data cleaning. Naveenatha, I spell the name if I right. Naveenatha ji, you have exactly mentioned pre-processing. Sanjay ji, cleaning. आपने भी सही बोला. So what we need for cleaning? जब हम cleaning करते हैं, when we clean something, we make it everything uniform. Rajni ji, we are going to make everything uniform now. Now just see this diagram, this table here. This is just a fictitious table which I created in my laptop. Just some name of person, Mark, Michelle, Andy. It has some years employed, some yearly income, position, sex, how many holidays they have taken, their experience in the industry. Okay, just see here, just for a glance, you can have this idea. Now, years employed are the number. Yearly income is a very huge number, 42,000. Okay, and holidays is 0 to 1, and the experience in the industry is 12, 4, 8, 17. Okay, just see this person number 11, Gil. Sanjay ji, you can see here, Gil.
Yes, sir.
Gil has years employed 1, okay, and his salary is 37,000. And see the topmost person, Mark. Mark has experience 13 and yearly income is 42,000. Okay. We need to normalize the numerical data variables. So what is being done in this example here, we have created one Excel file and we have read this file in the orange. We want to know which employees are more likely to go on holiday based on the yearly income, year employed in the company, and total years of the experience in the industry. So this data is a very tedious one. Suppose company is going to make a rule that some people will be allowed some holidays. Okay? For that purpose, we need to do a comparative analysis. For comparative analysis, we need to normalize the data. Okay? So normalizing करने के लिए there is another plot here. मैं आपको करके नहीं दिखाऊंगा. You can do practice later on, but I will be showing the results now. You have to plot the heat map here. ठीक है? Now just see here. आप देखिए the wage है. ठीक है? Salary, yearly income 42,000. This is a very huge number, and this year employed is a very small number. Computer doesn't know what is 42,000, what is 13. Computer just understand that these are by numbers. Okay? Computer is just a machine, and Orange is just a pre-processing tool, and it doesn't make a difference between 42,000 and 13. We have to normalize 13 and we have to normalize 42,000 so that they come in a some uniform range. For that purpose, there is another graph called heat map. So I'm not going to, this is assignment for you. What this heat map is showing you, this is the heat map. This is the non-normalized. Sanjay ji, you can see here, Rajni ji, 1 to 46,000. Okay, this diagram is a heat map without normalization. Now see the difference here. When we normalize the data, 1 become minus 1.97 and 46,000 become 1.68. So Sanjay ji, you can feel the difference. This is has a huge difference. 1 and 46,000 doesn't make a very good comparison. But when we normalize that value minus 1.97 to 1.68, this is a very small range. And according to the diagrams also, is very changing now. So this is a normalize the data. So sometimes your data values are very high. Any of my friends might have studied about histogram also. So what we do in histogram, the contrast of the image is very high on one side and the contrast of the image is very low on the one side. You make a contrast. We used to have old television sets at our home around 20 years back. So when we set the contrast, what we do all those things, we used to normalize those things. When we normalize anything, it provides some uniform range to all those values. So this is a normalization. You can plot the heat map and you find out where is the heat map, and then you see the difference here. Chaliye, I have another couple of 30 minutes left. Now I'm going to show you some research work to the image processing to the Orange now. See, whenever we do some research on a tool like image processing or bioinformatics, we have to deal with some data. So this data comes in shape and sizes. It can be simple spreadsheet or text. It can be video or it can be image. First, you have to install this image analytics toolbox here. I have already installed for you people. So this is the tool on which I'm going to show you now. The next task from here. This is the image. Here is the image analysis tool. There are a number of widgets, so we have to find sometimes it get merged in between. Yeah. All of you can see here, image analytics toolbox. Okay. Let's come back to the presentation now. So let's start with an example here. You can have a screenshot of this example. I will be using certain images here now, so that you can get those images from this link. I will be showing you some images of animals now, which I have already loaded in my image process, this Orange tool. Let's come back now. Just click on this import image. Okay. When we have to click the import images, we can, I have already chosen, you can browse it, or you can, this is my folder on which desktop Orange photos there, and I've selected domestic animals. Okay, just left click it and click on image viewer. Click here. Now click here. You can see here something. Anybody acknowledge me? Can you see the names of the animals?
Yes,
Yes,
Calf,
sir.
cat, this calf, cat, chick, cow, dog. Is it visible now?
Yes, sir.
Yes, sir.
Okay, thank you very much. Okay, what we are going to do, we are going to do image analysis now. We are going to do machine learning. See, the first animal is calf. Calf होती है गाय. My South Indian friends might be knowing cow को calf को गाय बोलते हैं. And second is cat. Cat किसके साथ resemble कर रही है? Cat resemble to, or you can say dog है. Dog किसके साथ resemble कर रहा है? Just make your imagination. This is roaster, and this let's have clarity. This hen and roaster, they belong to the same category. Sanjay ji, and turkey also same category.
Yes, sir.
Yes, sir.
Okay. Calf is the category of cow, and this milkha. Milkha is this American cow found in the mountain Alps. ये US में पाई जाती है ये गाय. तो this is a calf, this cow, and milkha. So we have what we are going to see here. I have shown you certain images of the animals here now. We have some 6 animals I have just represented here. What we are going to do now, we are going to categorize them. We are going to cluster them now. Okay. So first task is we have imported the images, and second is the image viewer. Okay. So come back here. Now just click on the data table. Sanjay ji, you can see here. What is data table here?
Yes, sir. It is representing different PNG files of all the animals.
Yeah, it is providing the size, width, and height. See, for image processing, these parameters are of no use. These are just some description of those images. We need some meta description. We need some more data about data. Okay, so for that purpose, we have got some useless description that Sanjay ji has also acknowledged that we have got the file name, location of the file, size, and image width. So that this data retrieved will not help us in the classification. We need some more image descriptors. So the next thing is that we have to make some connection. We have to train these images on a neural network. So that purpose, there is another widget called image embedding. I'm going to skip the theory part here. I'm going to show you the practical part.
Sir, the captions underneath the slide is also on, so it is
Pardon,
typing
ma'am?
whatever you're saying.
Pardon, ma'am?
Whatever you're saying is typed in the screen, sir. We are not able to see the full screen. The caption you have on, I feel.
Okay, let me see here. No, I just minimize it for 1 second. Can you say this screen?
Yeah, we can see the screen, sir. I want to say that whenever we are talking, no, the underneath whatever we are saying, it will be displayed.
Okay. Caption, actually, I have not enabled it.
Okay, sir.
I have
Thank
to
you.
see. I got your point, but I think that you want to see the captions. Okay. In which
Captions are on.
you want to on the captions.
No, it's already on. I don't know whether it's from
You
your
want
screen
it?
or.
Okay. I have to off it. Ma'am, you have to either off it because this will distract our talk. Okay.
Yes.
Captions are generally meant when the person is from the other part of the country or some other foreigner comes, so his voice is not very useful, or we are not customized to his accent. Okay, so let's come back to again this image. Can you see this import image viewer and data table? Okay.
Yes, I can see, sir.
Okay, next is image embedding. We're clicking to the image embedding. Image embedding is just see this theory here. What is this image embedding? This widget is the most important part of the entire image analytics tool. See, for classification and regression, we need some numbers only. And just in the initial start of when we started this diagram, Sanjay ji has mentioned that we got some height, width, and name. That data was not sufficient for the image analysis. So we need image embedding widget. What is image embedding does? It converts that image into a number of vectors. So just see here. You have to click here. When you click on the image, this is the embedder Inception v3. Forget about the details of the image embed. You can read it from Google. What is this? Actually, Inception v3 is a embedder provided by the neural networks, where it has already been trained on the real number of images, and then it extracts some useful information. Then again click this image embedding. to the data table now. Data table. Now Sanjay ji, see here, that same data which we already got. This was the previous data. Okay, this is the data. This is a data about data, meta description. What we are getting here, we are getting the features now. There are around, how many features are there? You can scroll it. Madam, you can see here, there are around 2048 features. So when you get a huge number of features, now this image, these features are having a very smaller decimal values, and for a human being to analyze these values is not a small cup of tea. I hope you understand all those things. So what was our purpose? Our purpose was to categorize those animals, that the cow should be in the cow category, and the hen should be in the hen category. Okay, well, let's proceed further. Now we have, earlier we have only the simple data. Now we have got the 2048 additional features. So this is called feature extraction. You all people know feature extractions are some mathematical descriptions of some values. So let's pass to another widget called distance widget. Connect image embedding to a distance. It will separate these widgets on the basis of the distance, and we cannot visualize the distance. For this we need hierarchical clustering. Click on the hierarchical clustering. Now see here, this is being categorized: cat and rabbit, hen and rooster. संजय जी, hen and rooster कहाँ पर थे? हम चलते हैं वापस इमेज व्यूअर पर. This is the hen. Hen कहाँ पर है? Hen, and this is rooster. Sanjay ji, do you agree with me?
Right, sir.
Okay, let's visualize something more again. This is goat and kid. Goat and kid देखते हैं कहाँ पर है? Goat देखिए कहाँ पर है?
On the right most.
Yeah, this is goat and this is kid. ये दोनों भी same है, almost
Right.
same family.
Right.
So this
Right.
is my dear friend, this is image clustering. अब आगे क्या दिक्कत आती है? अब next क्या होगा? Now the next thing is that this is your trained database. You download one image from the internet, okay, of any animal which resembles to this family, and if that family member of that animal is correctly classified by this workflow, which I have designed already, your result is good. Otherwise, your result is, or your algorithm is wrong. I hope you got the point. This is classification.
Please repeat this point.
Sanjay ji, suppose कीजिए, if you download one more image from the internet, any unknown image, say of a dog, ठीक है? Now, if that dog is being classified, dog comes in which family? Dog यहाँ पर, dog तो कोई family में है ही नहीं यहाँ पर, ठीक है? And if your system yeah, this is dog, ठीक है? If you download one bulldog image from the internet and you save that image again here, and then you try to find out that that particular dog which you have downloaded from the internet is again clustered rightly, it means that the workflow which we have created, this workflow which we have created is right, or otherwise your classifier chosen or your workflow which we have made is a bit inaccurate one. So this is the clustering which we have got here. You can find out again here. This is lamb and sheep. Lamb and sheep देखते हैं कौन सी family है? Lamb कहाँ पर है? Sheep.
Second row, sir.
Okay. Second row. Okay.
Fifth element.
Yeah, it is difficult to diagnose also. Okay, this is lamb. ठीक है? Sheep कहाँ पर है? Sheep. हाँ जी, एक मिनट सर. I just want to. Okay. Sheep कहाँ पर?
Third row, first element.
Third row. Third one.
हाँ, ये रही. ठीक है? तो see here, sheep and lamb, they are more or less similar one. ठीक है? This is clustering. Now somebody was asking something.
Yes, sir. I want to ask this orange, this software is comparing the picture attributes,
Yeah,
or it has
picture.
to do with names also.
नहीं नेम नहीं. संजय जी, प्लीज बेग योर पार्डन. नेम का कोई लेना देना नहीं. आप उसका नाम ABC भी रख दीजिए, कोई प्रॉब्लम नहीं है. फीचर, द मैथमेटिकल फीचर्स. Computer doesn't understand alphabets. Computer understand only the features, and features are just number for computers. Okay.
Right, sir.
I just want to share you something else also. This is the image classification for the architecture distortion, the mammograms, which I have done in my PhD work. In my PhD, we work on the mammographic images where the cancer prediction was done. So the abnormality which I chosen was architecture distortion. So I have created one folder here with two subfolders of 10 images of benign and malignant. Benign are the cancer one and malignant are the non-cancer one. Okay, so just see here. Now Sanjay ji, somebody else can see here. Is there any difference between is we were easily able to distinguish between, because the pictures of the animals were quite different now in that last one.
Yes, sir. Yes.
It was a drastic difference, but here, can you visualize the difference? Let me make it full screen. Can you visualize this difference? Benign, 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 benign. And if I scroll it down, these similar sort of images will be for the malignant one. Okay, just see here. These are benign images, and again I made the workflow here, but you can see here import images. I'm not going to do practice on my canvas now. So again the same procedure is followed. Import images, image embedding, and the data viewer. So by creating a workflow and applying the image embedding technique, so in this way I got those features, and I used cross validation and applied some other widget. I applied some other boost classifier and then performed logistic regression. While in the previous one, we performed hierarchical clustering. Now see the results here. The results are 50% only. You can see here area under curve is 50% and the number of folds is 10. The training set is 66%. See here the results are just 50%. Okay. I have taken only 20 samples, and out of 20 samples, I have made 14 as a training and rest as a test one. And the computer was not able to distinguish between the benign and the malignant samples. So in a research, whenever you do some analysis on the orange, try to have a huge database also, and the proper selection of those classifiers also, which are useful for your research. Okay. So this concludes the problem here. So my time is getting shorter, so I'm skipping the text processing here, but I'm going to give you one assignment now. I'm skipping the text clustering also here. So just for 5 minutes for some experimental setup for common classifier, which you can practice later on also.
Sir, sorry to interrupt you.
Yeah, ma'am.
Sorry to interrupt you. So just now you have shown some benign figures.
Yeah.
For those figures, how did you get that data table, sir?
Yeah, madam, that data table is my PhD research area. I've mentioned here also. Let me show the database also.
Okay, leave about the benign table. At least some previous figures you have seen some dot and for that also, how the data table was generated.
Ma'am, I have given. See here, this is the link which I have given also. This is the link from the images.
Yes.
Okay, and these images, I will be providing everything. Don't worry. So this is the. These images are from this sample, DDSM. You go onto Google and search DDSM. This is a digital database for screening mammography. Okay, you will get the images also. But GitHub.com also, that slide have the images also. So let me come to this common classifier, which can really help you.
No. What I am asking is, we will get the images, but for that images, which button, which icon we have to be pressed to get the data table?
For this purpose, this thing, import images, image analytics toolbox. Wait a second. So this import image, you have to click on the image, import image. You have to click on this browse button. This is a desktop. This is the image paint. See this image painting. I've seen here, image painting. I have clicked on image viewer and click here. Now you can see the images here. Yeah, this is by this way you are reading those images. Now see here, these are two types of painting called Manette and Monet. Okay, this was another experiment I have planned to give you as a homework, and you have to distinguish between the two painters' painting, Manette and Monet. They have so see here, the painting of Monet is something bit pattern different, while Monet is something different bit pattern. So this is also another diagram, another example for doing clustering of the images. Okay.
Now after these images, how will we get the data table?
This okay. I think you have missed, ma'am. You have to click on the data. Click on the data table. Okay. This is the data table now. This is the data table, and again, if you go for the image embedding, just watch my slides also. For getting these features, you have to connect import images with the image embedding and then image embedding is connected with a distance vector, and then distance vector will be connected with a hierarchical clustering. But it varies from image to image, algorithm to algorithm. So these are just two examples which I have shown you. So if you get the very good results from those hierarchical clustering, then it's fine. If you're not getting some good result, you have to choose another type of classifier.
Okay, sir. Okay, now, thank you.
Okay.
Thank you, sir.
So let's come. I want to give you one assignment for today. This is MTech level project for your student. You can do a comparative analysis of the various types of classifier. This is the small screenshot for that purpose, if you can see here. And this is a madam data set in the Excel form for your Excel purpose also. You can have a screenshot also for those. These are domestic animals zip, and this is the data set in the Excel form. There are a number of things to be learned about Excel, so to be learned about Orange, but considering the time factor given to me, I'm trying to tell you maximum things which I can tell. This is assignment for you people. You can have a screenshot, and if you really want to practice it, this will be a challenge for you. You have to collect the data also yourself. You have to classify the prediction of pneumonia from the chest X-ray images. Go to Google and find out the normal X-ray images. Okay, during this COVID time, there might be many images available on the internet. Whenever COVID happened, the pneumonia happens, and the chest X-ray get deteriorated. It get disturbed. So you will get around good number of images which are suffering from the COVID-19, and good number of images which are the normal ones. Okay. Now make a database of around 50 samples, where 25 are from the normal one and 25 are from the abnormal one. You have to load those images in the okay. And then you have to classify. You have already classified it, and then you have to find out the workflow. You have to find out which algorithm is suitable for that. Approximately, if you're getting the accuracy more than 90% by choosing some classifier like SVM or KNN or hierarchical clustering, that means your workflow you have created is a good one. Otherwise, the Orange will give you a result, but the accurate result will not be available unless and until you get some 90% result. So you have to predict and cross-validate it, and prediction and accuracy. So try to do this at your home also, this assignment. So this is the end of my lecture. This is the takeaway for your home. I've tried to discuss many types of widgets. I've tried to discuss some graphs also, some image classifier which I will discuss here, some tools which I've covered, some widgets which are very useful. And we have tried to cover visualization, pre-processing, normalization, subset selection. And still, if there is any doubt, I'm here. And thanks for listening. I hope the timeline was lesser as compared to the material or the things which we can learn, but considering all of you are quite mature one, and with just a simply introductory idea, you people can easily practice it at your home also. So that's all from my side. Thank you very much. Meenakshi ma'am, is it? Okay.
Thank you so much, sir. So participants, if you have any questions, you may write them in the chat box, or you can directly unmute yourself.
Although I've tried to involve many of the persons, thanks to Sanjay ji and many other names, Sandeep Kumar ji, and yeah, I will share PPT. Don't worry. Okay. Thank you. I hope the lecture might have done justice to you people, and you have enjoyed this lecture. This was not a traditional theoretical lecture, and the interaction with you people, around 10 to 15% were keep on interacting with me, and few person to name Sanjay, Shanta ji, and Bharti ji, Dhirendra ji. Thank you very much. For the network model, Abhirami ji, there is a network. I have stopped the sharing here. You have to do add-on here. Add-on करेंगे तो you will provide you will be prompted for a network prototype also. Okay. Thank you, Chandra ji. Okay. So Abhirami ji, your task will be easy done. But the thing is that I was not a person from the network domain, so I avoided giving lecture on that, because unless and until you are not customized to that particular tool, so it doesn't make a good sense. But you have a network tool in this also. It is similar, analogous to MATLAB.
Okay, sir. Okay. Thank you.
Okay.
Sir, hello.
Yeah.
Sir, can we do work on images like in a MATLAB? Some images can be colored images can be converted into gray images, or two images can be merged, and we can do some lots of work in images. So same thing
Generally
that can
image
be done.
conversion नहीं अभी possible है. Image feature, see, they have only recently discovered this toolbox, this image analytics toolbox. That's why the widgets are very less. You can see here there were only 4 to 5 widgets. The BioLab community is working on this aspect also. I have mailed also them some suggestions also, but right now the feature extraction is only possible. Feature extraction and feature classification. Conversion of images from gray to binary is not possible. For that purpose, you can use a tool like MATLAB and then load those images into Orange now. Maybe in the coming days, these sort of features will be also coming. But they have a good scope in text mining. If somebody is working on sentiment analysis, if somebody is doing on Twitter analysis, sentiment analysis, social media analysis, this Orange is a very good tool for those persons. As I was not, this lecture was of 1.5 hour duration, so I have to skip the text processing component. Otherwise, it will could have given you some very good idea about text processing also. Never mind, we will have some other day, some other platform. But text mining and sentiment analysis is a very now broad area of research in Orange. But for the image processing, it is just a beginner only.
Okay, sir.
Okay, I hope you might have enjoyed the lecture. See you once again on some other platform.
Yes, sir. Thank you so much, sir, for enlightening us with your knowledge. I think each and every participant really enjoyed your session and communicated with you, interacted with you throughout the session. Thank you so much, sir.
Thank you, ma'am. Thank you, Kamalji. Thank you, Dalibal Saab, if you are not there. Okay, thank you.